Hello, everyone. My name is Diana Wall, and I'm the director of the School of Global Environmental Sustainability at Colorado State University and the chairperson of this National Academy Study Committee. In my role as chair of this study and as moderator of this session, and I'd like to welcome everyone here on Zoom, those of you who are watching the live stream, and all of you who may be watching some days later. The task being undertaken by this committee of the National Academies of Sciences, Engineering, and Medicine is to explore the linkage between soil health and human health. The committee's full statement of task can be found on the study website, which is available at the link under Project to the right of the embedded video of this meeting. I'd like to give some sense of the format of this meeting today. I have a few housekeeping comments to make, and then I will turn things over to Carol Laney, who is the study director with the National Academies, to provide a brief overview of the study process. Following her presentation, I will introduce our speaker for today. To start, I want to note that this is an open, on-the-record session. Interested in individuals and members of the press have been invited to attend as observers. This is not a news conference, however, and we will not be entertaining questions from the audience. Reporters who would like to talk with members of the committee are kindly asked to touch base with Carol Laney, and also anyone who would like to submit a written comment or a document for the committee to look at should do so by clicking the Provide Feedback link on the right side of the web page. The committee's webpage. You can also subscribe for updates about the study by signing up to the National Academy's Board on Agriculture and Natural Resources listserv, also on the right side of that page. The committee will deliberate, I also would like to remind everybody that this is an information gathering process, a session. That is that the committee is in the process of assembling materials that will examine and discussing the course of making its findings, conclusions, and recommendations. Therefore, I ask everyone to be extremely mindful of the fact that the committee has made no conclusions yet, and that it would be a mistake for anyone to leave the meeting today thinking otherwise. Comments may be made by individuals, including members of the committee should not be interpreted as positions of the committee or the academies. In addition, committee members typically ask probing questions in these information gathering sessions that may not be indicative of their personal views. The committee will then deliberate carefully and thoroughly before writing its draft report. Moreover, once the draft report is written, it must go through a rigorous review by experts who are anonymous to the committee. And the committee then must respond to this review with appropriate revisions that adequately satisfy the Academy's Report Review Committee and the NAS president before it is considered an official Academy's report. This committee is composed of the 15 volunteer experts. You see the names listed on your screen, and you can find a short biography of each member on the study website. I'd now like to turn things over to Carol Laney, a senior program officer with the Board on Agriculture and Natural Resources at the National Academies and the study director for this activity. Carol will give you a brief overview of the National Academy study process. Carol?
Thanks, Diana. I just want to give a brief overview of the institution and, as Diana said, our study process to anyone who might be joining us today who's not familiar with the academies. So the National Academy of Sciences was established in a bill passed by Congress and signed into law by President Lincoln in 1863. And later the Academy was granted the authority to create the National Academy of Engineering and the Institute of Medicine, which in 2015 became the National Academy of Medicine. So today we're the National Academies of Sciences, Engineering, and Medicine, and the organization consists of those three honorific bodies, as well as the work of scientists across the country, inside and outside of the academies, who provide expert advice to the nation on issues involving science, engineering, and medicine. The mission detailed in the congressional charter is to investigate, examine, experiment, and report upon any subject of science or art whenever called upon by the government. We fulfill this mission in many ways, including through consensus studies like the one that we're discussing today. Just to say a little bit more about consensus studies as opposed to our other convening activities, such as workshops and forums. In consensus studies, they are conducted by committees convened specifically for a unique statement of task. The experts who serve on the committees do so as individuals, not as any representative of an organization or institution to which they may belong. They also serve as volunteers. Consensus study members review the state of the scientific literature to write a report that responds to the problem identified in the statement of task. The report will hopefully be used to shape sound policies, inform public opinion, and advance the pursuit of the scientific field in question. Each consensus study moves through a number of stages. First, the statement of task is defined through conversations between the national academies and the study sponsor. In this case, the U.S. Department of Agriculture's National Institute of Food and Agriculture is the sponsor for this study. Next, the national academies ask for nominations to identify individuals with the appropriate expertise and range of points of view to conduct the study. Each committee member should be comprised of individuals with not only the expertise, but also balanced perspective on the task questions. Nominees are appointed to a committee by the president of the National Academy of Sciences, Dr. Marcia McNutt, and committee membership is provisional until the committee has completed a discussion of conflict of interest, disclosures, and composition at its first meeting. After being appointed, the committee moves into its information gathering phase, which is the phase of the study that we're in now. Members of the public may also submit information to the committee for the committee to consider. As it gathers information, the committee begins to deliberate on its statement of task and how to best address it. And they will later begin drafting a report. When the draft is complete, the draft will be submitted to the report review committee, as Diana said, and will be sent to anonymous peer reviewers. The committee must then respond to the review comments. When the report review committee has decided that the committee has appropriately responded to the review comments, it approves the report for release, and the report is then delivered to the study sponsor and made publicly available. I just want to say a little bit more about the committee meeting process. When we meet with speakers, as we're doing today, we meet in open session, which means the speakers and the committee and staff are present, but also anyone who wants to listen in. If we meet in person, you can attend in person or watch the webcast. When the committee meets in closed session, it does so to deliberate on the statement of task and devise the best plan to address it. And in this case, only the committee members and National Academy staff are present. This is the general timeline for this soil health and human health study. This is our 8th public meeting. We'll be having another webinar tomorrow, also on the human gut microbiome. That's at 1:30 p.m. Eastern. And we have another meeting planned in person in DC next week. I anticipate that the draft report will be ready to review in the late fall and that the final report will be released in the spring of 2024. Here's the statement of task for this committee, and I'll give everyone a minute to read it over because it's lengthy. And if you go to the next slide, Kat. This is the second part of the task. And as I mentioned, the sponsor for this study is USDA's National Institute of Food and Agriculture. And the direction for this study came from a congressional appropriations bill to USDA. Finally, this is a screenshot of the study's website. You can find information on the left about our upcoming webinars, our past webinars, the statement of task, and the committee members' bios. On the right, you can find the link to provide feedback to the study or subscribe for updates about future news about the study. If you need to connect with the National Academy staff, here's my email address as well as that of my colleague, Catherine Kane. And with that, I'll turn it back to you, Diana.
Thank you, Kara. It's now my pleasure to introduce today's speaker. Bruce Hamaker is a distinguished professor of food science at Purdue University. He also holds the Roy L. Whistler Chair and is director of the Whistler Center of Carbohydrate Research. He has a master's in human nutrition and a PhD in food chemistry from Purdue and is published extensively in food science, human nutrition, and biochemistry journals. Bruce's research focuses on food carbohydrates with an emphasis on dietary fibers and the gut microbiome and glycemic and physiological systems. He also works in development-oriented research and applied programs in West Africa and East Africa and in Latin America. Bruce, welcome, and thank you so much for being here.
Thank you, Diana, and it's a pleasure to be with you all this afternoon. Let me share my screen here first. Okay, wait, sorry about this. Let me do this a different way. Let's see. Sorry, Diana, here. This usually is not complicated. Here, let me put this over here. There. Is that good?
I don't see anything.
You don't see anything?
I don't see your slides, no.
Okay. Oh, yeah. How about now?
Perfect.
Okay. You'd think I'd never done this before, right? Okay. Yeah, the topic of my talk today, and I want to leave plenty of time for questions because I'm not quite, totally sure what the committee is really interested in from, the aspect of food processing, nutrient density, which I don't have in the title, but certainly I'm going to talk about, and I can see the connection there, and the gut microbiome and health. I'm just as very quick disclosures. I and two other people have a startup company, Right Carbs LLC. I put this Choices International because it's a front of package nutritional labeling group out of the Netherlands, and of course we talk about nutrient density there. And it's a different version and type of front of package labeling system. And then I have some consulting right now with a prebiotic company, NutriLeads. And as director of the Whistler Center, it's an industry member-based center, so certainly deal with companies a lot in the food processing area and also do research with them. Whoops. Okay. All right, just very quick, what I'd like to go over in the next 30 to 35, 40 minutes is what does food processing encompass exactly, just quickly. A bit about nutrient density and changes in the gut microbiome. What is, very briefly, but what is a healthy gut and the role of dietary fiber? Of course, we work in this area of the intersection of dietary fiber with the bacterial community and related to gut health. I guess just to say, because I do have a nutrition background too, and also in a postdoc long ago, obviously, but we did get into in vivo studies long ago and do some animal and even clinical studies, but on healthy populations and then or cohorts. And then at Rush Medical School, have a very close collaborator and friend, Ali Keshavarjan, who in gastroenterology and the disease area. I want to give you some idea about dietary fiber types that are in the food and their classification, not going into too much depth, how we think anyway, and how really I think it's needed to think about fibers and the gut bacterial community. And then an idea that we've pursued and done a lot of research on about alignment of fibers to gut bacterial groups or even individual resident probiotic bacteria. And that's only reason I would like to say a bit about that, because I want to give you the idea of this high specificity interrelationship between dietary fiber, carbohydrate structures, and bacteria, and then end with a few slides on processing effect on fibers in the gut microbiota. So what is food processing? You know it, but it converts raw foods or agricultural commodities to foods that are acceptable, safe, ready to eat. There's criticism, obviously, and you all know that, about over-processing, or even it's now in the news a lot, ultra-processed foods. Myself, I don't object to the idea that some foods are kind of nutrient poor in the respect that they're not nutrient dense, right? But that they and that constant consumption may cause some health related problems, though I don't particularly like the definition of ultra processed foods. Kind of ends up you're stuck and can't move out of it in a way. But anyway, if you wanted to ask a question, I'd be happy to answer. So there's different degrees of processing, obviously, that include minimally processed foods like flours, milling of grains to flours. Of course, that removes a lot of fiber. For home cooking, for convenience, processed foods, and also going into convenient processed foods that have different ingredients added. So the way to think, I believe, about the gut, the microbial community related to processed foods is really focusing on dietary fiber, also phytochemicals, phytonutrients such as polyphenols, and even protein. And I'll talk a bit more about that later. So there's a variety of processes, don't need to get in. There's very into all those simple ones, but then there's very disruptive food processes, disruptive, I mean, as far as what they do to the food material. And a good example would be extrusion processing. You all know extruded foods, extruded breakfast cereal, ready-to-eat breakfast cereal products, snack products, expanded cereal or starchy-based products that are crispy to eat and enjoyable to eat, but the process has changed the components to a good degree. For instance, the starch usually is readily available to the body, and even usually those are high glycemic kind of foods. But the fiber, again, I would ask you to think about that a bit differently, because fiber that enters the gut is food for the gut bacterial community is not necessarily changed to the worst, can even be changed to the better. Okay, so, go to what is called ultra-processed foods are really foods made of a variety of ingredients and also have a lot of times disruptive processes, mechanical processes like extrusion or other kind of processes. I know you're going to hear in I think a couple of days, Cara said, a whole lecture on the gut microbiome. You'll hear, I'm sure, a lot of things that I can't talk about, but just to say it, that the GI tract, of course, has is populated with bacteria from the mouth all the way to the rectum. But if we just look at the colon, we have a big increase in bacterial population numbers, And those numbers are greater than 1 trillion bacteria in the colon. And it's about 1.3 times the human somatic cells. And the bacterial population community structures differ certainly from the mouth all the way down. And in the colon, there's the bacteroidetes. I know that the word has changed there. Anyway, I still am calling it that, at least for the meantime. Contains Bacteroides, Prevotella, which I know are bacteria that are present in high levels also in the soil microbiome, also in the gut. Clostridium groups, here we have 4, there's other ones too, but here's 4 and 14 are important for health. And these are just some, Bifidobacterium Enterobacteriaceae. There's many others. So what is this business of nutrient density and how might it affect the gut microbiome? I think I just put a really simple definition that Adam Drunowski, who's researched and written a lot about this concept of nutrient density, has in a 2014 paper in American Journal of Clinical Nutrition. Foods that supply relatively more nutrients than calories would be a nutrient dense food, and a food that has lots of calories but relatively few variety of nutrients would be one that is not nutrient dense. Excuse me. So again, nutrient density for the gut microbiome means basically dietary fibers, carbohydrates that escape digestion, which are all of them basically, except, starch being the major one in foods, but also other starch products that are ingredients that are added to foods, lactose, sucrose, and a few other kinds of alpha glucans that can be digested in the upper GI tract, in the small intestines. Also phytochemicals, the best example, and perhaps the most important group there is the polyphenols related to the gut microbiome. I will say that while a lot has been learned, and I'm going to show you that there's the science has come quite far in the area of dietary fiber structures and how they affect the bacterial community in the gut. Phytochemicals, I'd say, and polyphenols is in an early stage of research, but we do research in that area too. I'd be happy to answer any questions in that area. Protein, generally thought of as negative. It has negative consequences in the colon, but it also is used by some bacteria to produce like favorable, desirable short chain fatty acids metabolites like butyrate. So, generally though, it's for a healthy gut, it's thought that protein levels should be on the low side. But also it's worth saying that in current diets that with a lot of emphasis or certain diets for certain groups of people who consume a lot of protein and a lot of plant-based proteins, more of that protein ends up in the gut. Gut, I'm saying, is really large intestine. So Justin Sonnenburg at Stanford and his wife Erica published a very well-cited paper back in 2014 in Cell Metabolism that shows an idea about microbiota diversity, or diversity of the the bacteria in the gut related to the diet. And what he calls, I don't know if you can see this well, sorry, but what I call dietary fiber, they call microbiota accessible carbohydrates, or MACs, MACs. It's the same thing. But if you have high dietary fiber, or MACs, you get more microbiota diversity, more metabolic outputs. That's in the form of short chain fatty acids shown here. And there's in general a relationship with that and protection of Western, chronic non-communicable diseases related mostly to low-grade inflammation and sometimes poor barrier function. In industrialized populations, generalization, right? But a decrease in dietary fiber can correlate to a decrease in microbiota diversity and metabolites, short chain fatty acids. And there's a relationship that has grown over time that's quite convincing with certain diseases and health conditions, maybe not the sole cause, but a significant cause of a number of, or related to, maybe I shouldn't say cause, associated with a number of, again, chronic diseases. Put that in the perspective of how we've evolved or have come from early man to present times. They have this nice figure here of how microbiota diversity was probably quite large in the hunter-gatherers and in early agriculture. I read a paper some years ago in a European Journal of Clinical Nutrition. It was by a paleobotanist, guy named Jim Leach, and he proposed that he had one picture of evidence in the paper. The one data picture was like fossilized feces, and it had a lot of fiber attached to it. So what he proposed was in early man that dietary fiber intake was up around 100 grams, even perhaps more than that a day. And of course, our average consumption in the United States is around 15 grams. Anyway, that fiber, even though maybe it wasn't all fermentable, but still a large, say 30% of it was fermentable. That's twice what we take in today on average. But as the industrialized age and processing was introduced and processed sanitized food has been introduced more recently, where a lot of fibers have been removed from those foods, that microbiota diversity has gone down. And the only way to get that back up is with high fiber diets, diverse fiber diets, and species reintroduction. Really, this says the same thing, but in mouse studies that high fiber diets maintain a diverse population. That's because all the different fibers in that diet are feeding different bacteria and in that competitive environment, satisfying everybody. But low fiber diets don't do that. And over generations, there's study, and there's been more than one in animal studies, have reduced diversity. And even though you transfer it to a high fiber diet, those different generations, you can't recover bacteria that's not there, right? So low gut microbial diversity is linked to certain disease states like metabolic syndrome, type 2 diabetes, inflammatory bowel disease, and colorectal cancer. So diet has a big impact on the gut microbial community, as you would imagine. And I went to a meeting a couple of weeks ago in Denver, a really great meeting called ISAP. It's on prebiotics and probiotics. And a microbiologist stood up and said, the question was asked, what is the contribution of diet versus somebody's genomics on the community structure, on setting community structure? He said it's about 80 or more % diet. Wow. And here's a paper that's often cited to just show that there are big differences in the gut microbial community due to diet. This was a study done in Italy, and here's an African children, their gut microbiomes were examined in Burkina Faso, and this is in Florence, Italy. And you can see that from the phyla standpoint, Bacteroidetes, Firmicutes, that there is a big difference between the two as far as community structure and predominant bacteria between the two. In Africa, leaning towards Bacteroidetes, in Europe, leaning towards Firmicutes. That's not maybe just on the surface saying this is bad, that's good, just showing that difference. So what is a healthy gut? And these are the two main things, low inflammation and high barrier function. Chronic systemic inflammation and poor barrier function is related, as I've mentioned, to a variety of non-communicable chronic diseases. And diet can lead to a healthy gut microbiome, may either prevent or reverse them. I mentioned the metabolites of dietary fiber fermentation or carbohydrate fermentation in the gut, the main ones being acetate, propionate, and butyrate. There's been a lot of interest in butyrate because mechanistically it acts with the localized immune system at the colon level to reduce inflammation. It's been shown, we've published a study even in this area that on improvement of gut barrier function. And it's interesting that butyrogenic bacteria these commensal clostridial bacteria found in the Firmicutes phylum are located near the gut mucosa, up near the epithelial cells of the lining of the colon. Just a little bit more about these Clostridium bacteria in suppressing inflammation. I won't get into the weeds here, but there is a crosstalk between the immune system and the bacteria out here producing really the butyrate. And you can see Commensal Clostridia kind of residing, not wholly, but the majority in the mucosal surface. They have these kind of bacteria. You may know, some of you I'm sure do know it because they exist in the soil. And I learned quite a bit about how they get into matrices and all from the soil microbiome literature when we first got into this area. And they have these cellulosome-like appendages coming out from the cell surface. Down here, if you can see that, the bacteria can switch out enzymes and binding proteins and even eject this scaffold into matrices to digest out small oligomers that are then turned into sugars for energy for the bacteria. So it goes along with an idea from the whole food side that whole foods have more fibers that are cell wall matrix entrapped dietary fibers, and that those fibers from whole foods give a preference or have a more of an alignment towards these kind of clostridial commensal bacteria. We did a study with our partner in Chicago, Ali Keshavarjan, recently, and it was the paper was just published in February, where we, using some ideas we had about fibers supporting different groups of bacteria and different key core groups of bacteria in the gut to make a prebiotic fiber mixture or mixtures that would support the important bacterial groups in the gut. And here is a heat map with the red showing the promotion of bacteria. And we used 4 different fibers. I mean, it's not a mystery what they are, but just didn't want to get into the detail. Those are commercial fibers. And you can see that they complement each other. This was just published in Nature Communications. And then the interesting thing, the clinical study was done on Parkinson's disease patients. And Parkinson's disease, it's known that the gut microbiome, that there's dysbiosis, out of balance bacterial communities. And in 10 days of 20 grams a day of tolerable fibers, these are also, there was a reduction in proteobacteria, that phylum that tends to have, or it has some members certainly that are pro-inflammatory. Serum zonulin decreased, meaning that gut barrier function increased, and localized and even brain inflammation was markers of both calprotectin and this NFL protein were reduced. And interestingly, also in a group from Caltech that we were a sub on this project, Sarkis Mazmanian, they showed in Parkinson's disease, they have a mouse model called the AZO knockout, that the mice have Parkinson's disease-like symptoms, that the prebiotic, a similar prebiotic mixture, complementary mixture that was designed to support key core bacterial groups, showed, I don't want to get into any detail, just to say that it had a marked effect on even motor movement outcomes, and also blood and at the brain level microglia, which in Parkinson's disease and this mouse model also become abnormally shaped with thickened appendages and this thick center part. But the prebiotic fiber, not reversed it, but made that normal from, these were young mice that were fed the fiber versus a control. So very quickly, I'm going to talk about dietary fibers, just to let you know, and I think you all probably do, that there's a big variety of different kinds of dietary fibers in nature and in the foods that we eat. And that includes sugar composition, polymer size, linkage types, the linkages that combine these sugars, and branching distribution, and also related to those fibers being soluble or found inside a cell matrix, like I was talking about earlier, and are insoluble. There's kind of a misnomer out there, and with a number of people, and I would say a number of people even in the food industry, that soluble fibers are good for the bacteria, they're fermentable, and insoluble fibers are bulking agents and basically not fermented. That's just not true, as you can see from some of the things I said before. I'm going to skip this, getting into a little bit more detail on dietary fibers, but only to say that in each fiber group, whether it's oligosaccharides in this slide to polysaccharides, one can find structurally simple fibers, in this case, like fructooligosaccharides and inulin, that are just the fructose sugar, and they have one linkage attaching them. So you can imagine from the bacterial side that there are many bacteria that's an easy fiber to break down to sugars, and many bacteria have the machinery, the genes encoded in their genome for that particular kind of carbohydrate. As you get to structurally complex, whether it's oligosaccharides or polysaccharides, generally there are less bacteria that can utilize those kind of complex structures. And it's easier to conceptualize how one could identify or design fibers that would promote those specific kind of bacteria. And to say also that each bacteria would have maybe 15 or 20 different cassettes for different kinds of cassettes of genes encoding, what's necessary to break down different types of carbohydrates. So I'm going to jump now through this and say that from the regulatory side, that the classification of fibers really it's not FDA's fault, but fiber's fiber, right? There's to a lot of consumers, it's just important to get fiber into the diet. Doesn't really matter what kind. But from our perspective, it's quite important what kind that is, or at least to get a variety of different kinds of fibers. On the nutrition fact panel, you can put one thing on that differentiates or classifies fibers into soluble versus insoluble, though that's a voluntary statement, not mandatory. We tend to think of fibers in terms of a spectrum of fermentability, fermentable versus all the way to non-fermentable. There's also prebiotic and non-prebiotic. And just to say, though this is not perfect, but to think about a fiber fermentability spectrum where there's highly fermentable fibers like inulin and phos, again, that have one type of sugar, one type of linkage, so easy to get at. And bacteria ferment those really quickly, can cause even abdominal distress, into chemically complex polymers and even oligomers where rate of fermentation goes down. This is the soluble portion. Increased tolerability goes this way, and then into a range of different insoluble fibers going from, most of them would be fermentable to some degree, and a few would be very hard to ferment. I'm going to jump. And I'm just going to, in a few minutes, 5 minutes, I'm going to give myself to finish. So I'm just going to tell you very quickly, and I put too much slides, and so I'm going to jump a bit, but our own research, because I think it's instructive for one thing, where the science is now. And we're not the only ones, doing research, but we're particularly in this area, but in this particular topic, we're kind of the ones that are deep into this. This is the idea that fermentable fiber structures can be found to align with particular gut bacteria, or as I mentioned in that Parkinson's disease study, groups of bacteria. And it goes on this premise that there's so many types of dietary fibers, and any given bacteria would have different strategies to get food, to get sugars. So some are going to be going, they'll have the ability to go after like inulin that a lot of other bacteria are going after, but they may also have a cassette, or called polysaccharide utilization loci, of genes encoded for a difficult to digest and utilize fiber. If you can find that, you've aligned a fiber to a bacteria that could specifically support that bacteria. And here's an example of that. So this is a genus abundance plot, and this is time going this way from left to right within these panels. All I want you to do is look at this gray one bacteria. It's a genus Anaerostipes. It's a clostridial bacteria in that Clostridium 14A cluster. It started out, it's butyrate producing, and it started out at 0 time at less than 0.5 relative abundance. And in 24 hours, it went above 20, even in this case, above 25% of relative abundance. It's like that the fiber was so specific to that bacteria that it was basically feeding that particular bacteria. Little bit of change in that structure, you lose that. So my apologies to jump around a little bit. We've taken this idea into the idea, if you get a specific kind of relationship between a fiber and a bacteria or bacterial group again, can you make it work consistently across people? Because if you have that bacteria, everybody on this call, and you can identify that structure that's more specific to that bacteria, you should get the same response in everybody. And here's a really just example that is easy to see. This is inulin, which FOS and inulin are basically the same thing. Inulin, I should say inulin is the most added fiber in processed foods in the US, Europe, and also Japan. But it's this very simple one. And this non-specificity in 10 different donors, these are 10 different individuals, you get a lot of different responses. This is called a beta diversity plot. I don't need to get into anything about that, just to show that the bacteria are responding differently because their community structures are different. We published in 2020 an idea about this idea I've been talking about low specificity, high specificity fibers, and that you could get consistent response. And with this high specificity fiber I showed you for Anaerosis stipes, that Clostridial bacteria, you can see you get a bigger response and the same kind of response in almost all donors. This particular donor didn't have that bacteria. And this just shows that in another form, that the response also is more robust and intense, say for fructooligosaccharides, inulin, and bifidobacteria, which is another target. We've also figured out a protocol to find those matched fibers. And this is for another bacteria that there's a relationship with health, with inflammation, localized inflammation. And we've identified a fiber through a protocol that specifically supports that, again, across donors. Okay, so finally, what role does processing have? I mentioned that it can reduce intrinsic dietary fibers, and oftentimes in processed food, that's replaced with added single fibers like inulin. Not that inulin's the devil or anything, but that's just that what has happened. And that may or may not support a healthy gut in the long term. I would say no single fiber is going to support a healthy gut in a low fiber diet. It may increase the soluble and insoluble fiber ratios, which is not necessarily desirable in the long term, and may increase or reduce, this is processing, fiber fermentability. So I'm going to, in the last two slides, just show you that it's possible to, with processing, actually improve the value of an insoluble fiber that's not that fermentable. You know, just to comment on polyphenol composition, certainly processing can change that composition, reduce it, the amounts, but even change the composition. But its effect is really not known at this point. So I don't want to get into the data, but just to say, I think this is from last year. We published a paper called Boosting the Value of Insoluble Dietary Fiber to Increase Gut Fermentability Through Food Processing. And the goal there was to take insoluble, not that well fermented fiber and kind of explode it with a food processing technique. We used just a high energy microwave in this case with some enzymes, but we kept the insoluble nature of that, increased fermentability in orange, and increased butyrate levels. Not that easy to see here, but we doubled butyrate levels from the untreated fiber. So I think I don't need to get into that. And I'm going to break for questions.
Thank you very much, Bruce. That was really very informative for me, who never thinks that much about these gut microbiome. We will now take questions from our committee, and we have a few minutes for questions. Who's got their hand up first? Aha, Sarah.
Hi, thanks a lot for that presentation. That was wonderful. I just wanted to ask if there's any more that you could tell us about what you're finding with phytochemicals, because I think there's a big relationship between soil health and plant phytochemical content, and so curious your perspective on it.
Yeah, we just completed a study on phytochemicals where we looked at 4 different types of phytochemicals and 4 different kinds of fibers and that kind of factorial design you can imagine. And it's quite interesting that that any given phytochemical will respond differently depending on what fiber is there, for one thing. But then also, we saw some big differences in the effects of phytochemicals on the effect of certain bacteria. I'll just give you an example. One of the phytochemicals was a mix from chocolate, from cocoa, right? And in that particular phytochemical mix, there was, no matter what the fiber type, in that case, it increased certain bacteria. And actually, they happened to be desirable bacteria like that Faecalibacterium prausnitzii was promoted in every case. I think I would say it really takes a good effort to understand what's going on at that level of phytochemicals. But I do think there's a high specificity of phytochemical structure to affect. A lot of studies, because it's, I think, just in the beginning, that's been going on for a while now, but still, the initial studies in phytochemicals with polyphenols and all were really looking at what just sort of a gross effect. If you eat this much phytochemicals, what happens to the gut microbiome? There is an effect, but then figuring out what exactly is going on, is that positive or negative? We saw some interesting positive effects. That's not the definitive study.
We have some more questions? I know we do, but in the meantime, I'll ask one, Bruce. You mentioned the Clostridium and it's being also in soil, and that there have been studies on that linkage to the gut microbiome or the gut bacteria. Is that a growing subject that is going on right now? Is this link between, that you know of, that are studies that are looking at like nutrient density and the types of criteria you have? Is that a ongoing, more energetic side of the looking at linkages on how do you get the best microbiota in your gut?
Mm-hmm. There's quite a few groups that are looking at those commensal clostridial bacteria. Mm-hmm. I don't know how much they're looking at the soil, microbiome literature, that particular, the clostridial bacteria. But as I say, it was really useful in our own research, and found this big Clostridia, soil clostridial. You probably know that there's a handbook, and it's a very thick one. I didn't read the whole thing. But there, I would say it's a growing interest, Diana, in how those bacteria can be supported through the diet, because there's a thinking out there, and in our minds too, that modern diets and diets that are not nutrient dense, perhaps, and there's a low fiber or replacement of fibers with single, I'll use inulin again, that kind of replacement fiber is not really supporting that group of bacteria, the commensal Clostridia. And so, that's of interest to a number of people. I hear it around.
Let's see if we have any more questions here.
I could say one other thing, because at this ISAP, pro and prebiotic meeting in Denver that I went to. It was interesting to me that the probiotic group has always been bigger than the prebiotic, but that's changing a bit. But it was interesting to me that it was said in the last day session, the overview of the different discussion groups and all, that probiotics, their positive effect on human health. So, introduce bacteria that may or may not be, or that are not already part of the community, or they're through the oral route, that their action seems to, usually not, was the message I got, but I could say often not, related to their becoming part of that community. That it is not easy for external, of course, there's examples, right, of bacteria, environmental bacteria becoming part of the gut, community, but it's not a very easy thing and so that the probiotic effect, like for, say, a lactobacillus, strain, is really by taking it every day and it having a particular effect at the mucosal surface.
We have a hand. Kate Scow is on the committee.
Hi, thanks so much for such an interesting talk. I was wondering, because we think about this in soil, is there ever like amendment of pro plus prebiotic where you're providing the kind of fiber that those particular organisms would use in the gut, or does that thing just go to hell when it hits the gut? And
you mean to
like
the prebiotic plus probiotic, is that right?
Yeah, right, to provide the organism with its substrate that it would be particularly good at, to try to nudge it.
I mean, exactly. That's a great question. And that's, we think that with the approach we've taken of, aligning fibers and identifying fibers that give that preference to that bacteria, that can be done. In the grocery store or pharmacy, if you go and you get, you buy a probiotic and it says plus prebiotic, that's just any kind of prebiotic. I think probably there's confusion at the consumer level on that. But right now, that's not the aligned. I could say that in some cases, maybe it's okay that bacteria can use this particular fiber, but they wouldn't know that when that fiber goes into the gut, whether it's eaten by other bacteria before that bacteria can really use it. But I think what's on the horizon is go ahead.
Maybe it's putting them together or something so they can get to where they need to be, and then
it's really I think this is where the field is heading. It's going to happen for sure.
And I just wanted to say something really, really fast, is that often, like I've worked a lot in bioremediation, and there's this prebiotic analogy going on there. Do you need to add organic compounds to help move it along, or oxygen, or do you need to add the organism? And always, you usually need to apply both. But often, you don't need the organism. What you need to do is change the conditions, is provide the prebiotic components so that indigenous organisms can do the work. And I don't know if that's, maybe that's how people think about it. Yeah.
That's really fascinating. I didn't know that. It makes good sense. It's the same. I would say it's basically the same.
We're getting close to time. Do we have any more questions that we want to ask Bruce? Well, Bruce, I just want to say this is really, to me, very informative, and I think to the committee too, and it helps a whole lot with where is the field going. So I appreciate, on behalf of the committee, the time you gave us, and I'm sure if we have any questions, we could get back to you through Kara.
Yeah, feel free. Thank you. Enjoyed being with you. Bye-bye.
Thanks,
Bye.
everybody.